Alan, we might as well talk about him a little bit because he is our seminar speaker today. Alan Lazarus and I have known each other for way too many years to admit to. He and I have sat on many streets in the world at various meetings and watched the people go by, and he will testify to that. He started out at McGill University doing a PhD in micro and immunology, and then he did a postdoc at Banting and Best at U of T. His first appointment in faculty was as a staff scientist at St. Mike's. Then I guess would also have been accompanied with a faculty position at University of Toronto. He later saw the light, was offered a stupendously lucrative position with Canadian Blood Services. I will not tell you the year because that is approximately when I joined Canadian Blood Services, and it wasn't even called Canadian Blood Services then. It was called the Canadian Red Cross Society. He is now appointed with many dubious distinctions. One, and most importantly, is senior scientist with Canadian Blood Services. He is professor of medicine at University of Toronto, and he is the co-director of the Toronto Platelet Immunology Group, and he is the co-director of the St. Mike's Hospital Transfusional Hematology Research Theme. And without further ado, I will leave you with Dr. Lazarus to tell us whatever he'd like to speak about today.
Well, thanks for that wonderful introduction, Ed. I don't know if I should talk about anything I want to talk about today. Maybe we'll stick to the science. Hopefully you guys can see my pointer. So the title of my talk is Innovative Strategies for Replacing IVIg in Autoimmune Disease, the Role of Fc Receptor Blockade. So here are my disclosures. So in this talk, we're first going to talk about IVIg, look at its mechanisms of action of IVIg, at least in ITP. Then I'm going to introduce you to the concept of Fc gamma receptor blockade or Fc receptor blockade. We're then going to look a little bit at recombinant Fc multimers that are in development and hopefully we'll be seeing in a clinic one day soon. Then we're going to talk a little bit about antibody methods of inducing Fc receptor blockade and some of the problems with adverse event activity with that strategy. Then we're going to talk about our work on monovalent Fc receptor blockade. And then finally, we're going to talk about a new molecule to block Fc gamma receptors that was accepted for publication yesterday. So hot off the press. So we're going to be talking about IVIg, and for those of you who may not be familiar, IVIg is basically antibodies collected from the plasma of healthy blood donors. This is what a typical IgG molecule looks like. We'll be talking quite a bit about the Fc region, so that's the part of the IgG that interacts with things like Fc receptors or complement. And then, of course, there's a part of the IgG that combines with the antigen, the Fab'2 region, that we're going to talk a bit about, but not quite as much. And IVIg is basically a collection of antibodies, and that's pretty much what it is. So if you're going to talk about IVIg and how to replace it, you have to look at a particular immune disease, and we're looking at the disease ITP, which now stands for immune thrombocytopenia. You could see it's had a number of name changes over the years, but we've always managed to keep the ITP part. And ITP is a bleeding disorder that's caused by a low platelet count. So it's an autoimmune disease, and what ends up happening in ITP is you end up getting IgG antibodies that bind to the surface of the platelet. Once the platelet is sensitized with IgG antibodies, that allows the platelet to bind to Fc receptors on macrophages, located usually in the spleen, sometimes in the liver. And then the macrophage phagocytoses or engulfs the antibody-coated platelet, and that results in a low platelet count. And when you give individuals with this disease IVIg, about 70 to 80% of them, the platelet count goes up. So IVIg is a concentrated preparation of human antibodies. It's produced from the plasma of thousands of blood donors. Some of those lots are as high as 100,000 blood donors in product that comes from the US. It is as pure a population of IgG as we can possibly make. It's exceptionally expensive, and it's a very limited resource. So in Canada, we only collect enough plasma to make about 17% of what we need, and that number keeps going down all the time. And so it's a real worry. Can we replace it with something recombinant made in the lab so that we could use IVIg for things that are absolutely essential? So that's a little bit about what IVIg is. So IVIg is now used in many, many different autoimmune and inflammatory diseases. And the very first publication showing its effects in an autoimmune disease was this publication by Paul Imbach, and here's a picture of him later in life. And in this particular paper, what he showed is that IVIg was able to increase platelet counts in children with the autoimmune disease ITP. And since then, it has been used in a multitude of different diseases, and IVIg use continually increases on average 10% per year. And so, of course, we're getting into trouble. So even though IVIg was used predominantly in ITP as well as in immune deficiency, which is completely different, still today, or actually this isn't today, but this is a review in 2015 by Andrew Shee, where he showed that at least in this audit of IVIg in Ontario in 2015, that out of the 178 new requests for IVIg, the majority of them came for hematology. And if you do the breakdown by disease, most of them were ITP, followed by secondary immune deficiency, so a lot of not indicated, Guillain-Barré syndrome, and then you can find your favorite disease. And if it's an antibody-mediated autoimmune or inflammatory disease, there's a good chance that IVIg works in it, or at least has been tried. So we use a lot of IVIg. How does it work? How does it increase platelet counts in ITP? How does it make Guillain-Barré disease better? That's a $1 million question, and unfortunately, I don't have a clear answer for you. But here are the theories of action of IVIg, and I've given talks just talking about the mechanisms of action of IVIg. It could easily fill up an hour. I'm not going to do it, but I'm just going to present you here with the list of mechanisms that have been proposed. Some of them kind of dubious, some of them have much more substantiated. And we're going to look at a very non-scientific approach as to how IVIg works by using Dr. Google. So, and I did this 2 days ago. So if you go on Google and you put in IVIg, mechanism of action, ITP, and 2024, because I was interested in the most recent publications, you end up with this publication here in 2022, suggesting that the mechanism of action is that monomeric IgG in high dose IVIg causes blockade of activating Fc gamma receptors by saturating them on immune cells, attenuating the pathology. If you then look at what other people ask, kind of some interesting questions, probably because I asked this question from Canada, this Canadian Blood Services thing popped up, and I agree with it. Mechanism not yet proven. We really don't know precisely how IVIg works. And then the last one here is a Medscape article saying that IVIg works by blockade of macrophage Fc receptors. It is considered the primary mechanism of action of immune globulin in persons with ITP and other autoantibody-mediated diseases. So Fc receptor blockade is one of the mechanisms that's been postulated for IVIg. And that makes sense. If you look at the list of diseases, and this is a list of diseases that was put together a number of years ago, but here's a list of diseases at the time. It was known that IVIg worked in these diseases, or very likely worked. Some of the diseases are very rare. So for example, autoimmune neutropenia, I think it's quite clear that IVIg is beneficial, but there's not enough patients to ever do a clinical study. And aside from the hypogammaglobulinemia, which is immune deficiency, so IVIg works by a different mechanism, pretty much all of these involve IgG antibodies as part of the disease. And so it's reasonable that IVIg might work by Fc receptor blockade, or at least an equivalent type mechanism. So when we look at the theories of action of IVIg, even though there are many theories that are still very worthwhile considering, we're going to focus in on Fc receptor blockade because what I'm interested in doing, or one of the things we're interested in doing in our lab, is trying to replace IVIg with something recombinant. I mean, IVIg consumes 50% of the budget for transfusion in Canada. So it's a very expensive product. And if we could find a way to replace it, I think that would be beneficial. So in terms of how could IVIg block Fc receptors, we talked a little bit about the pathophysiology of ITP, and probably other diseases work very similarly where you have an antibody interacting with an Fc receptor. So one possibility is that when you're getting an infusion of IVIg, you're getting IgG antibodies from thousands and thousands of individuals that are dissimilar to yourself. So it's very likely that you're going to have the IgG infused into you, and it is going to bind to other things. It's going to bind to antigens. They could be soluble antigens, as I've pictured here, or it could be the surface of cells. It could be antibodies interacting with other antibodies, but you're basically going to form these types of immune complexes. And these immune complexes are very capable of binding to and blocking or activating Fc receptors. And so that's one way to explain how could IVIg work to block Fc receptors. Okay. So how have people tried to take advantage of this? Well, work in our lab actually showed back in 2005 that you could substitute immune complexes for IVIg. Nobody was really interested in that idea, but a few years later, what people decided to do was just take the IgG Fc region and make it in such a way that it forms multimers with other IgG Fc regions. And so these Fc multimers, they're the part of the IVIg that actually is what probably triggers the activity. For example, these could bind to and block Fc receptors, but we don't require a blood donor to have these made. And so a company called Glicknick I think it's kind of in the Washington, Baltimore area, has come up with something they call the Stratomer that looks like this. It's a bunch of IgG Fc regions. This product works in a number of different mouse models of autoimmune disease, ITP, a mouse model related to multiple sclerosis where IVIg could be helpful. Another product called Hexagard that was designed by someone by the name of Richard Please from Liverpool. What his idea was to take the IgG Fc region and fuse that to the IgM tailpiece so that you end up getting pentamers or hexamers. And this molecule also works in a number of different diseases where IVIg works. And then finally, a product from Momenta Pharmaceuticals in the Boston region. They then formed a collaboration with CSL, and they have an IgG Fc that looks like this. And all of these actually can substitute for IVIg effects in various mouse models and other models of autoimmune disease or inflammation. So for those of you who are interested more on sort of these IVIg substitutes, there's a really nice review article in Autoimmunity Reviews published in 2019, where Adrian Zucker from CSL Behring actually looked at the next generation compounds, so the recombinant Fc multimers. I talked about a few of them. There are more. As you can see, they are in various phases of clinical development. We still have a ways to go. One of the good things about these molecules is that they do seem to work, but the problem potentially is the adverse event activity. So these multimers are likely going to cross-link a lot of different Fc receptors and drive adverse event activity. Whether or not we can get around that or not, I think will be a question. I'm not going to talk about FcRn-targeted therapeutics, not today. And then, of course, there's the direct approach of actually targeting Fc gamma receptors directly using some targeted therapeutics. And here I'm going to talk about our work. You can see it's characterized as Canadian Blood Services, monovalent, what they called monovalent 3G8 variant. I don't know that I would have called it that, but we're going to talk about that for the rest of this seminar. So the idea that the FC receptors are playing a major role in ITP and probably some of these other autoimmune diseases, and that perhaps rather than giving IVIg, one could just block the FC receptors, is an idea that Clarkson and Jim Bissell and others actually thought about. And what they did is they had available an antibody that binds to and blocks a human FC receptor. There are three different major activating human FC receptors, FC receptor 1, 2, and 3. And an antibody at that time was available that bound to and blocked FC receptor 3. And what Clarkson did is actually showed in a New England Journal of Medicine paper way back in 1986 that this antibody could increase the platelet counts, at least in one patient with ITP. It looked quite promising. They took the antibody, they converted it from a mouse antibody into a human antibody. They modified the FC region by removing the glycan. In the absence of the glycan, it's not pictured here, IgG antibodies cannot meaningfully interact with FC receptors or complement, and so they thought this might work better than the original mouse antibody. They tested it, and I think it was about 16 patients and a few abstracts in blood. The antibody worked well. It increased platelet counts. The problem with the antibody is that it caused horrible adverse event activity, and that pretty much killed the idea of using an antibody to block FC receptors for quite a long time, until a super talented postdoc by the name of Ben Yu came to my lab, and he was interested in picking up FC receptor blockade, but sort of doing it in a different way. And so the idea that Ben had was, instead of using the antibody that actually has the ability to bind an FC receptor in two different places and thereby cross-linking the FC receptor and actually causing activation, that perhaps instead of doing that, we could use either an FAB fragment or something else. And of course, an FAB fragment's not going to work because the half-life is only, I don't know, it's minutes to hours. And so what we did is we teamed up with Bill Sheffield from McMaster and also Canadian Blood Services to design a stabilized kind of equivalent of an FAB. What we did is we took albumin and made a single chain fusion protein consisting of albumin as well as sequences from two FC receptor blocking antibodies. The first one was an antibody that blocked mouse FC receptor 3 so that we could do in vivo work in a mouse. And we also used the same sequences from the papers I showed you before, an antibody called 3G8 that binds to and blocks human FC receptor 3, and that we fused to human albumin. And so we tested both of them, one in vitro and one in vivo. So this is what the data looked like. So if you take mice and you inject them with a antiplatelet antibody, they develop thrombocytopenia. And as you can see, so this is a 2-hour time point. Sorry, it got cut off. But when you inject a mouse with antiplatelet antibody, within about 2 hours, you develop a thrombocytopenia. We're only giving a single injection of antibody, so eventually the platelet count will come up. And as you can see, when we injected our monovalent FC receptor 3 blocker, you start to see an increase in the platelet count. And at 80 micrograms per mouse, which is actually a very low dose, it's only about 3 milligrams per kilogram body weight. So when you give this dose of antibody, it actually blunts the thrombocytopenia. So you don't need an intact antibody. The monovalent fragment worked quite well to increase platelet counts. Well, that's great, but what about the adverse events? So that's the next thing that Ben looked at, was adverse event activity. And here I'm going to show you body temperature. We did other measurements. You can look at Ben's paper if you're interested in. And so what you do is when you take the mice and you dose them with an antibody, so the first antibody we used is an antibody called 2.4G2, which is a mouse version that binds and blocks FC receptor 3. And then we give our antiplatelet antibody and just look at temperature changes in the mouse over time from this injection. We can see actually that the temperature dropped quite alarmingly within 30 minutes of giving the full-length antibody, even though the antibody had been FC region deglycosylated. So there's really no FC activity here. But probably the cross-linking by the two FABs is what's driving this drop in temperature. And then so what we did is we used the mouse monovalent, and as you can see, the mouse monovalent, as I showed you before, it blocks the FC receptors, but there's absolutely no change in temperature. So this allowed us to block FC receptors without causing this major adverse event activity. Well, what was the cause of the adverse event activity? And what Ben did is a really cool experiment where he took basically the same dose of the 2.4 G2 mouse serum albumin construct, and then followed that with an antibody to the His tag that we had put on this molecule for purification. And when you do that, you could see he recapitulated a drop in temperature. In fact, it's even worse than the original antibody. So this showed that the reason why we're getting these adverse events was likely because of cross-linking the FC receptors in the mouse that's driving all this adverse event activity. So the answer for us at this time was that you can block FC receptors successfully and increase platelet counts, at least in mice. And as long as you do it with a monovalent strategy, so no cross-linking the FC receptor, you're not going to get this major adverse event activity. Great. So we decided to go ahead and move to the next level. And of course, the question then was, does monovalent 3G8, so the original antibodies used in these studies, does monovalent 3G8 work in vivo, or do we need to design a new molecule? And just to give you the answer, the short answer, the answer is no. Unfortunately, the 3G8 albumin fusion protein didn't seem to work well enough to do anything in the mice. And so, well, and you'll see that data soon. But what we needed to do was design a new molecule from scratch. And so we did that. So what we did is we formed a collaboration with Admari Bio, which is located in Vancouver. I think they now have offices in Montreal as well. They're an accelerator. We put together a grant with themselves, myself and Bill Sheffield, and we decided to make our own molecule. So this involved immunizing mice with recombinant human FC gamma receptors, and then we used those mice. We got the DNA from the spleen, and we did phage. Well, we didn't do phage display. Admari Bio did phage display, and came up with phage that are able to bind and block FC receptor 3. Human FC receptor 3, and we looked for example, amino acids that would be not friendly to us, and came up with this molecule, beautiful name, 17CO2, which we went forward to use as a potential therapeutic. And we took those sequences of the variable and light the variable, heavy, and light regions of the antibody, and made 3 different molecules. We made the same as we showed before, an albumin fusion protein. So you have the heavy and light chain, single chain with a human albumin, and a His tag for purification. We made a one-armed antibody. So it's an antibody that only has one arm. So this would also allow us to monovalently block FC receptors, but not use the albumin structure. And then we just made a regular standard antibody as a comparator, and asked which does this actually work? So the first experiments we did is we took our 17CO2 albumin and we compared it with the 3G8 albumin that we had been working with for a number of years, and asked, does this actually work better? And so we looked at binding to macrophages that express FCR3 or CD16, and you can see our 17CO2 molecule binds much better than the 3G8 albumin. Natural killer cells that only express FCR3, they don't express any other FC receptors, at least that we know about. Again, our molecule binds quite nicely to natural killer cells and better than 3G8. Interestingly enough, they both bind equally well to neutrophils. Neutrophils have a different version of FCR3. It's called FCR3b, and rather than a transmembrane receptor, it's not transmembrane and has a very small number of amino acid differences, and our molecule binded about equally with 3G8 to the neutrophils. We tried to avoid binding to this receptor, but it just was not possible. So it works well to bind. I don't know about you guys. Okay. And so what we needed to do is test it in a model. Of course, we're not going to test it in humans. We can't test it in mice because mice express mouse FC receptor 3. And so what we did is from Jeff Ravitch, we received mice that have been genetically modified such that they do not express any mouse FC receptor. So mouse FC receptor 1, FC receptor 2B, FC receptor 3, and FC receptor 4 have been genetically deleted. And Jeff put in human FC receptor 1, human FC receptor 1. You can see that the monocytes express it. They also express human FC receptor 2A. They express human FC receptor 2B, so the human inhibitory FC receptor, the receptor we're interested in, FC receptor 3A, and even the neutrophils express the neutrophil FC receptor 3B. And for those of you who are interested in platelets, the platelets had the correct expression of FC receptors, only expressing FC receptor 2A. And so we have these mice in our facility, thank you, Jeff, which we are now using to go forward with. So the question is, using these kind of very adulterated mice, so these mice, they've got multiple genetic deletions, they have a lot of human material put in, what actually happens in these animals? And so looking at adverse event activity with the FC gamma receptor humanized mice, we tried our FC receptor blockade. And what you can see is that when you use the 3G8 down here, which is the same antibody that failed in the clinic from an adverse event activity, again, in these mice, it triggers a drop in body temperature change. The same is true with our 17CO2 albumin when it's expressed as a full-length antibody. Again, we've deglycosylated both of these, so the only thing that it can do is cross-link FC receptors, and again, that also drove adverse event activity, but our 17CO2 albumin, no temperature drop. So same thing is true with the 3G8 albumin. So when you monovalently bind the FC receptors, there's no adverse event activity, at least not this adverse event. I'm sure there'll be others, but we haven't found any. What about as a therapeutic? Does it actually work in the mice? So here we've used, again, the FC receptor humanized mice. And first what we do is we take the mice and we dose them with an FC receptor blocking reagent. We then wait 2 hours, then we induce thrombocytopenia. And for this, we use a rabbit anti-mouse platelet serum. We then wait, and then at 4 hours, we bleed the mice and we do platelet counts to see whether or not the experiment was successful. So if you look at untreated mice, their platelet counts around 1,000. Mice have a much higher platelet count than humans. If you take the mice and you give them an antiplatelet antibody, they develop thrombocytopenia. And actually, all of the groups here have been injected with the antiplatelet antibody, and you can see the only place where you see an increase in the platelet count is mice that were dosed with 17CO2 albumin. So our new monovalent FC receptor 3 blocker actually increases the platelet count in the mice. And this is what I told you before about the 3G8 albumin does not work. It doesn't have the power to increase the platelet count. Albumin doesn't work. The various isotype controls don't work. And what's interesting is that if you actually take 3G8 as a full-length antibody or the 17CO2 as a full-length antibody, they also did not increase the platelet count. I'm not going to go into the details. If you're interested, I'm going to put the paper up at the end, and please take a look at that paper. The reason why the full-length antibodies don't work is one of the side effects that they induce is actually thrombocytopenia on their own. So when you cross-link FC receptors, this actually drives the thrombocytopenia in the mice. So monovalent blocking of FCR3 actually works nicely in this humanized ITP mouse model. In addition to doing the 17CO2 albumin, we also, as I said before, we used a sort of a monovalent FC receptor, one-armed antibody called 17CO2 IgG1-OA. So it's a human IgG1 formulated in such a way that it only can bind the antigen once. And we asked, does it work? So it actually works nicely. So here's our ITP model. Here's normal mice. Here's mice induced with ITP. When you give the one-armed antibody, it also increases the platelet count. So you don't need the albumin fusion protein. The one-armed antibody works well. But when you look at the adverse event activity, it's even though the drop in temperature is not as bad as the full-length antibodies, they went down to around 32 degrees, we're still getting adverse event activity. We're not 100% sure why. When we look at this molecule, the way we purify it is with protein G chromatography. That gets us up to about 98 to 99% purity. So it could be the 1% of full-length antibody. So there is going to be sometimes a little bit of full-length antibody that's made. We've tried putting a his tag on here to do purification, and still, no matter what we do, there's still a little bit of temperature drop. So for some reason, I think the one-armed antibody is going to be something that is going to be a little bit more difficult to work with. So I'm very happy to say this work was published, well, not published, but was accepted for publication yesterday. The title of the paper is Human Fc Gamma Receptor 3A Blockade Inhibits Platelet Destruction in a Humanized Murine Model of ITP. This work was done by Lazaro Gil Gonzalez and Kevin Doyan Wan. They're the co-first authors. And also our collaborators at Admari Bio, as well as a lot of other collaborators that helped put this forward. So I don't know when it will actually come out, but it was accepted yesterday. So to conclude, we still do not know precisely how IVIg works, but Fc gamma receptor blockade is considered to be one of the primary mechanisms of action of IVIg. Certainly in ITP, it seems like that might be the way it's going. There's probably other mechanisms involved, but based on that, a lot of us have looked at Fc receptor blockade. There has been significant development in IgG-based recombinant therapeutics showing that Fc multimers, which, by the way, block Fc receptors, can mimic IVIg activity. We don't know that that's the mechanism of how they work, but that's what people talk about. I did show you that monovalent Fc gamma receptor blockade can increase platelet counts in a humanized Fc gamma receptor-based ITP model without triggering major adverse event activity. And if anyone has $5 million to take this to the next step that you're not doing anything with, give me a shout. I'd be happy to talk to you. So I'd like to thank you guys for listening. I'd also like to thank the members of the lab who've done some wonderful work. A lot of the work that I talked about was done by Lazaro Gil and Kevin in the lab. We were also helped by Bill Sheffield early on, Jim Burak, George Siegel and Rich Burak from Rochester, who helped us out with macrophages, Dr. Uli Sachs from Gießen, Germany, who gave us access to ITP sera. I'd like to thank Yuxin Shan, who did a lot of the molecular biology in our lab to drive this project. And thank you guys for listening. And that's it.
Thanks for that terrific talk. You're so articulate and never fail to be so, Alan. Are there any questions from the audience? Well, Alan, you know, I'm always wondering, you and I always talk about control experiments. I'm wondering if in your single arm antibody, whether isotype controls, like some irrelevant antibody, trypanosome specific antibody, is going to have similar types of effects, whether that FC is doing anything.
Are you talking about as a therapeutic or inducing the...
No, I'm talking about in your control experiments, whether or not that's helping to explain part of the effects that you're seeing.
You know, it's a good question. So we haven't really looked at different specificities. I mean, of course, we use isotype controls that are matched to the antibody as closely as we can, but they're of no relevant specificity. So whether or not we were to use antibodies that had an irrelevant specificity, but not the one we're interested in, is a really good question. And actually, in another paper that we have out there, reviewers have asked us that question. So it's a good question, certainly. And there's always a possibility that, you know, just picking another isotype control with an irrelevant specificity is going to help fish that out.
And are you expecting this wonderful new therapeutic that you've invented to spare the need for plasma donation, as well as save the Canadian taxpayers' dollars, or just the former?
Well, so, it's probably right now we're only 17% self-sufficient for IVIg. So more than 80% of the IVIg just comes from paid US donors. So I don't think it's going to reduce the demand for plasma, but hopefully it will reduce IVIg usage. That's the idea. The idea is to reduce IVIg usage. So that's why we're trying to look at a mechanism that's as close to IVIg as we can get to, and hopefully this helps spare IVIg use. That's the goal.
Thank you. Caitlin, you've got some questions from people online?
Yeah.
Thank you.
Ed and Alan, thanks very much. That was great, Alan. It's Doug Morrison here at BC Children's, and I have a couple of questions, one relating to mechanism and the other relating to dosing. In terms of the mechanism, I'm just wondering if FC receptor blockade plays a role in some of the other autoinflammatory conditions that IVIg is used for. So in I guess peripheral nervous system disorders, central nervous system disorders, and a whole gamut of other autoimmune and autoinflammatory disorders. Do you think? Do you have a comment on the mechanism in those conditions?
That's a really good question. And unfortunately, our knowledge of immunology in diseases is just inadequate. So in mouse models, so for example, you're talking about peripheral nerve, so for example, EAE, which is a mouse model related to multiple sclerosis. There's really another model called EAN, which is closer to CIDP, where IVIg is used a lot. That's not a workable model, but at least in EAE, we know that the FC receptor gamma chain is needed for full disease development. Is that because of FC receptors? It's not it's something we're actually planning on doing. So we got somebody who's an expert on EAE, and we're going to see whether or not we can block FC receptors and whether or not that could be part of the solution. But unfortunately, we're way behind in understanding the pathophysiology of most autoimmune diseases. We know that the antibodies are there, but it relies really on mouse or other animal models you're not going to do something to a patient to explore whether or not FC receptors are involved. So I think we haven't gotten there yet. Some of the diseases, we're working on some other models with these antibodies. I'm not ready to talk about them yet, but sort of as we're going, we're asking, okay, are FC receptors involved in this particular type of inflammatory response? And can blocking it be helpful? So it's kind of a bit of a slugfest that way.
Okay, thank you. Can I just follow up quickly with a question about dosing, which is something that we wrestle with every day when in the clinical use of IVIg, and it's the evolution of 2 gram per kilo dosing, for example, in the induction sort of portion of the treatment of some autoimmune conditions, and then typically 1 gram per kilo monthly. But there's another condition, neonatal alloimmune thrombocytopenia, wherein the mothers are given, or the pregnant woman is given very substantial doses, like 1 gram per kilo weekly of IVIg, and I think it's in some studies has been that sort of aggressive regimen has been effective, and it's kind of a standard regimen now. And I'm just wondering how what your thoughts are around the dosing piece, and are you by escalating the dose in that fashion, are you inducing greater catabolism of the IgG molecules. And just your thoughts on that, please.
Yeah, so FNATE's actually it's one of the things we're looking at, and hopefully we'll have a paper published pretty soon on this molecule in FNATE. So it is one of the things that we're looking at, but FNATE's a little complicated with IVIG because of the neonatal FC receptor. So, IVIG works in adults, and we've shown formally that IVIG does not require FcRn as part of its function in increasing platelet counts in ITP. We published that back in 2012. I think around 2012. So we don't think that IVIG uses FcRn as part of its mechanism. But in FNATE, where you now have whatever that IVIG mechanism is, plus you've got FcRn that plays a role. So there's some work from Hey-Yuni's lab showing that IVIG might be working in a manner dependent on FcRn in a mouse model related to FNATE. It becomes more complicated. So when you start getting to the point of how do you do your dosing, and what do you look for, we still don't know what the primary mechanism of IVIG is. And now in FNATE, you have the two. You've got FcRn plus whatever that primary mechanism is. So it's a little bit too complicated to really weigh in and say, okay, a dose of A or B or C is going to be helpful. I mean, I remember somebody, actually it was Jim Bissell, who told me years ago, he said, I don't think IVIG works by FcRn because you could see it within 24, 48 hours, and FcRn takes days before you start to see a drop in IgG levels. But I think in that situation you're talking about, it's too complicated.
Thank you.
All right.
Thanks a lot for joining us, Doug. Jordan, did you have some questions?
Yeah, I had a question. Hi, Jordan Shavit from the University of Michigan, where I think you visited us about a year ago. Nice to see you again.
Yeah, good to see you.
Great talk. Thank you. I have a question about the side effects of IVIG and wonder if you have any insight into the mechanism. As you probably know, some patients will have aseptic meningitis as a side effect of IVIG. I've seen some patients who don't get it at all, and then other patients who get it with every single dose, their first dose and then ongoing. Is there anything does it have anything to do with FC receptor, or is it a different mechanism?
You got me there. I have I've got no idea. Nobody's really looked at it. So, in the old days, IVIG had a lot of aggregates in it and they kind of cleaned it up. And I think, I don't know, like what happened to the aseptic meningitis in the old days when they had a lot more aggregates versus today. That would kind of give you a little bit of an insight as to whether or not it might be down that road. Or, the problem with IVIG is if you look at a list of things that it binds to in vivo, the last time I looked, which is maybe about 10 years ago, the list had 27 different things on it, including HLA, B cells, T cells. I mean, it binds everything. So it's going to be a real tough one. I'd be happy to talk to you about the aseptic meningitis part because it's really interesting. I don't know, if you've gotten any insight there, but especially if you're seeing those kind of things, it would be interesting to sort of explore it a little further and try to find out what's actually going on. Are there more? One of the things that we think that happens with IVIG is it's forming immune complexes. And it would be really nice if there's somebody out there with patients that they're giving IVIG to who's interested in this kind of stuff, to look at immune complex formation in the patient after getting the IVIG. I think there's some clinical tests for rheumatoid factor, and asking, does that correlate with anything? Because if it correlated, then it would be interesting.
Okay. Well, I'll send you a follow-up email and maybe we can chat about it some more.
Yeah.
All right.
Great.
Thank you.
Thanks.
Thanks very much, Jordan. Anyone else have some questions for Alan? I see Dr. Conway waving his arm.
Hi, Alan. That was great. So it remains a puzzle. I don't understand why you guys can't figure this out. I had a question. Have nanobodies against FcRn ever been developed or used?
I mean, there's quite a few reagents that target FcRn in the works. So, you know, there's FCs, there's antibodies specific for the FcRn. So there's 3 of them. So Argenx has something called efgartigimod. There's quite a few FcRn agonists or antagonists, I guess antagonists out there that seem to work well in autoimmune disease. So it's licensed, for example, in some diseases, and they're now looking at ITP. A nanobody in particular, I don't know.
Thanks. Seeing more arms waving. Oh, one more. Yes.
I have one more online from Georgina. She's asking, does your albumin conjugated antibody have potential for treating rheumatoid arthritis?
That's a really good question. I mean, it would have potential to treat inflammatory arthritis. I don't know if it would likely be successful in rheumatoid arthritis, and then that's getting down into the weeds. The thing about rheumatoid arthritis is that you have rheumatoid factor, and rheumatoid factor is very similar to an immune complex, which is what I think that IVIg is doing when you inject it. So I think in rheumatoid arthritis, it's not likely, but some other types of arthritis where there's no rheumatoid factor, it's a possibility that it could be helpful. But that won't be for a long time.
I have now got the all clear from Caitlin that there are no further questions for you, Alan, from the world. So thank you so much for joining us and giving a really interesting talk. Lots of questions were asked from all over the place. I hope to see you soon, and certainly in Saskatoon. And with that, I wish you a great rest of the day.
Hi, everyone. Thank you.
Thanks very much.